Newsflash, climate change exists, and as a society, we are past the point of ignoring the consequences of our inaction. Now, we know the consequences are substantial, but the consequence I am most interested in is how climate change negatively affects agriculture, and by extension, global food security. As drier, hotter weather becomes more frequent, agriculture struggles, which significantly strains freshwater resources and increases food insecurity. Simply put, climate change disrupts the specific conditions plants require to grow, causing significant agricultural loss, which we can't afford. In this video, I'm going to focus on one solution which can mitigate this particular consequence. The method? Gene modification. With new technologies like CRISPR, we can create crop plants that can withstand the impacts of climate change, which is essential as food insecurity soars and traditional farming methods fail. So, how do we do gene modification? Let's start by exploring our options. We could, for example, modify nutrient density. In the 1990s, scientists developed rice with increased vitamin A to fight childhood blindness. Another example is increasing pest resistance by adding immunity genes, which is critical as global warming is creating new habitats for invasive species, which devastate crop production. Of course, gene modification can also be used to increase drought resistance, which is the example I'll explore today. So, the bottom of plant leaves are dotted with tiny openings called stomata, which are crucial in gas exchange, but also release small concentrations of water. This water loss is worsened in long periods of windy, hot, and/or dry environments due to equilibria exchanges. Droughts are becoming more common and sustained, which devastates crop yield and strains our freshwater resources. However, it is possible to reduce water loss through adjusting stomatal density. So, how do we do this? Well, researchers have pinpointed one gene group called epidermal patterning factor, or EPF, which is the most promising for density manipulations. There are 11 families of EPF peptides, and researchers have discovered that by adjusting different variations of these families, they can reduce the density of stomata on a leaf, which significantly increases drought resistance. Success! Alas, many problems remain. Firstly, it takes time for these crops to be integrated into fields, and even longer for them to enter grocery stores. Secondly, the total success of one plant must be repeated into other crop plants to create real impact. Lastly, the stigma surrounding the safety of genetically modified plants is a significant hurdle to be overcome. Well, this was a very broad and brief introduction into the captivating intersection of agriculture, gene modification, biology, and law. I hope you enjoyed this presentation, and I encourage you all to keep investigating and discover something that intrigues and excites you, just as I found this.